Now we are actually going to talk about the structure of these contractile proteins which are present inside the filaments. So first we'll talk about the thin or the actin myofilament. The average diameter of a thin myofilament is around 70 angstrom. So this is the diameter of the actin myofilament. Each actin myofilament, that is the thin myofilament we were talking about, they are made up of 3 types of proteins. The major being the actin, then the tropomyosin, and there is troponin. So these are the 3 types of proteins which are present in the actin myofilament or the thin myofilament, which we can say. There is actin, there is tropomyosin, and there is troponin. This is a picture in which they are arranged, the 3 types of proteins are arranged to form the complete thin myofilament. So now let's study the structure. Each thin filament is actually made up of 2 F filamentous actins which are helically wound to each other. So now what happens, there are these molecules of actin which are known as the G actin. So this is one G actin. Now G actins, they under the influence of magnesium ion, G actins, they polymerize and they form a long chain which is known as the F actin, also known as the filamentous actin. So this is one chain known as the F actin, that is filamentous actin. Similarly, to form the thin actin myofilament, 2 F chains are present. So this is one F chain completely. This is one F actin we can say, or a filamentous actin we can say. Similarly, there are 2 F actins which are present like this, and they are helically wound to each other. So we can have a look of these filaments over here. So this is one F actin filament. This is one F actin, and this is the other F actin which is present. They are helically wound to each other. So this is the arrangement of actin. Then each F actin, as I told, is a polymer of monomeric G actin or the globular actin, which polymerize, which form the F actin under the influence of the magnesium ions. The 2 F filaments are helically wound to each other. Now, apart from this, we can see there are other type of proteins. This is the other type of protein which is known as the tropomyosin. So this is the tropomyosin, the brown color filamentous structure. Now, there are 2 additionally present tropomyosin molecules. So 2 filaments of another protein known as tropomyosin, they also form a double-stranded alpha helix and they run close to the F actins throughout its length. So we can see over here, these are again 2 tropomyosin filaments which are also helically wound to each other. So this is the second protein. The tropomyosin is a fibrous molecule that attaches to the F actin in the grooves between its filaments. So the areas where there are grooves between the F filaments, it is there where the tropomyosin attaches. Then comes the next type of a protein that is known as the troponin protein. So you can see over here, this is the troponin protein. These are the troponin protein which are present at regular intervals along the length of F actin and tropomyosin. These are actually a complex proteins. Why? Because they are formed of 3 polypeptides. These are distributed along regular intervals on the tropomyosin specifically. So troponin is made up of 3 types of subunits. The very first being, we can see over here, this whole is the troponin complete. This is troponin and it is made up of 3 subunits. This is TPT, there is TPC and there is TPI. So this is the TPT, this is the TPC and this is the TPI. Now let's see what is TPT. TPT is that part of the troponin, it is that subunit of troponin which binds to the tropomyosin. We can see over here, this T stands for the tropomyosin to which it binds. Along with this, it also binds to the other two troponin components. So we can see over here, this is TPT. TPT is going to bind to the tropomyosin as well as to the other two components. The next comes the TPC, which is known as troponin C. It is a calcium binding polypeptide, right? It binds the calcium. So the C over here is actually standing for the calcium. And this binding to the calcium itself is actually considered as the start of the contraction process. So in the next video, where we are going to talk about the whole contraction process, that is the sliding filament theory, there we will see what exactly is the role of TPC molecule. Then comes the last of troponin is TPI, also known as troponin I or the troponin inhibiting molecule. This is that molecule which inhibits the interaction of actin as well as myosin. That is, it is going to actually mask that site of actin to which the myosin will join and initiate the contraction. So during the resting phase, the masking of the actin site, that is the myosin binding site on actin, is done by this component that is known as the TPI component of troponin. So these were the three types of proteins which form up the thin filament of the muscle. Now we are going to talk about the thick, also known as the myosin myofilament. The thickness of the myosin myofilament, it is around 150 angstrom. And each myosin filament is actually considered, it is also a polymerized protein which is made up of many monomeric units, that means many repeating units, which are known as meromyosins. So we can see this is a picture of a meromyosin. This is one meromyosin altogether. These ways, meromyosins, they keep on joining together to form the complete thick myofilament, also known as the myosin myofilament. So this is just one unit, and in this way the meromyosins, they keep on joining together to form the myosin filament altogether. Now, each meromyosin has two important parts. We can see one is the globular head with a short arm and the other is a tail. So you can have a look over here. This is the globular head along with a small area over here, and this is the complete part that is known as the tail. This is the tail. This is the short arm, and this complete area is known as the globular head. So now, the former is also called the heavy meromyosin, also called the HMM, that is this complete area having the head as well as the short arm. This complete is known as the HMM, also known as heavy meromyosin. Whereas this area, that is the tail area, is known as the LMM, also known as the light meromyosin. This whole area is the HMM, known as the heavy meromyosin. We can see over here again, the former is known as the heavy meromyosin, also known as HMM, and the later is known as the light meromyosin, that is LMM. The HMM component, we can say, it projects outwards at regular interval and it forms the angle from the surface of the polymerized myosin filament. And it is known as the cross arm as well. So this complete area, this complete part, the head and the short arm, that is actually known as the cross arm also completely. And at regular intervals, it keeps on projecting out from the whole length of the filament. Suppose this is the length of the filament altogether. So the head, the cross arm portion will be projected this way towards the whole length. So like this, like this, like this, like this, they will be projected outwards. It is this part actually which is going to bind with the actin filament and it is going to initiate the contraction. So the globular head is an active ATPase enzyme and has a binding site for ATP and active site for the actin as well. We can see over here very clearly. These two are the actin binding sites. That is, it is from this site that the myosin filament is actually going to bind with the actin. And it is at this particular site, as we can see over here, these two sites are actually the ATP binding site, which are going to bind to the ATP and facilitate the breakdown of ATP because of the presence of enzyme ATPase in it. So this was the structure of light as well as the heavy myofilament.
To buy these books, you can go to www.cengage.co.in, or you can also find the links in the video description given below. Thank you.